Today we're talking about cell structure and function. This is in your textbook in Chapter 3, Sections 1 and 2. Before we get started, check to make sure that you have your notes organizer downloaded. This has a place for you to write down all of the important information and really helps you to focus on the key points from the notes. So make sure that you have this to include this handy chart of organelles on the back. That's going to be a really handy study tool for you later on in this unit. So if you need to pause the video, go ahead and do that now and then come back when you have your notes organizer. So to begin with, we're going to talk about some of the people that were instrumental in our understanding of cells today. And there is a place for you to just briefly write down the contributions of each of these scientists that we're about to talk about. The first one is Robert Hooke, and he was the first person to actually use the word cell, this word that we use all of the time today. And he did that by using a very primitive form of a microscope, meaning it wasn't as sophisticated as the microscopes that we would have in class. It could only magnify things about 30 times. But he was able to thinly slice through cork, which is from the inner bark of a tree. And he noticed, as you can see in this picture here, this is his sketch of what he saw. There were very regular box-like structures. And they reminded him of the rooms where the monks would live, which were called cells. So he started calling them cells, and the name stuck. The second person to be aware of is Anton Von Leovenhook, and he did not actually invent the microscope, but what he did was he was very, very skilled at grinding his own microscope lenses, so he was able to greatly improve upon the microscope, and you can see here on the slide, he could magnify things up to 300 times, so he was able to see much more clearly the fine level of detail of some of these organisms. And he really was the first person to discover that there's a whole world of microbes in pond water. He would look at lots of different types of organisms, and he discovered single-celled organisms. He even discovered that blood had cells in it, and he also was famous for studying sperm cells because people did not know what was in sperm, and so he was able to use the microscope that you see here on the screen to actually study those fine details. Now, you might be wondering how that thing even works as a microscope. And I like this diagram here. This picture shows you a replica of a Leeuwenhoek microscope. And notice how small it is. So that small size really made him able to consistently make those lenses so precisely that he was able to get this magnification. And so you can also see on the slide some of the sketches that he made. He really was just amazed at the diversity of single-celled critters he found in pond water. Next up, we have a trio of scientists. They all had their own contributions here, but together their work has led us to what we know today as the cell theory. So we have Matthias Scheiden, Theodor Schwann, and Rudolf Virchow. And they each studied different kinds of cells, but part of science is to share what you have learned with your colleagues. And so in sharing what they have learned, they were able to come up with the cell theory that is really a unifying theme in biology. This is a fundamental way of understanding living things throughout all of biology. So let's look at that cell theory in more detail. There are three core principles that are a part of the cell theory. The first one is that all organisms are made of cells, and this is so fundamental in biology that we learned it as one of our characteristics of life. The second principle is that all existing cells are produced by other living cells. Now, what that means is that cells don't just spontaneously appear out of nothing. People used to think that, for instance, if you just left hay in your barn and it got wet, then it would spontaneously produce mice, not realizing that adult mice would have gotten in there and they would have had babies, and that is why you had so many mice. Same thing with meat. They thought that the meat that spoiled when they noticed maggots crawling in the meat, they thought that they spontaneously appeared rather than realizing that they came from the eggs laid by flies that had landed on the meat. So this was really a huge step forward in understanding how biological organisms work, to know that there had to be some pre-existing cell that gave rise to that new cell rather than have it spontaneously appear. And the third part of the cell theory is that the cell is the most basic unit of life. What does that mean? Well, it just means that the cell is the smallest part of an organism that is still going to show all 7 or 8 characteristics of life, depending on how you count them. So you've probably heard before that there are 2 major kinds of cells. There are prokaryotes and eukaryotes. And I do like to always break down the meaning of the different word parts. So here you can see the prefix pro in biology typically means before. The prefix eu means true. And then the karyo part, the karyon, actually literally translates to kernel. But what that kernel refers to is, of course, the nucleus. So it's a large kernel-like structure inside of a cell. And that's where the words prokaryote and eukaryote originate. Now, the difference between these cells is going to be that prokaryotes are very simple. They don't have a true nucleus. So if you think of the word part, before nucleus, right? Prokaryon, before nucleus. And they also lack other membrane-bound organelles. We're going to talk about a lot of organelles in this presentation that are unique to eukaryotic cells, and prokaryotes pretty much lack all of them. Now, eukaryotes, on the other hand, will be much larger and more complex than prokaryotic cells. The DNA is contained within a nucleus, and they have many other membrane-bound organelles inside of those cells. So just as a point of comparison here, let's look and see what structures are common between prokaryotes and eukaryotes, and then we'll focus more on the structures that differ between them. So first of all cells have these same characteristics, and the first one is that they tend to be microscopic. This is something we will talk about a little bit later this semester, why cells have to be small. It has to do with their ability to transport things across a membrane. But that does lead to cells being very tiny. Now, another thing that all cells have in common is they are surrounded by a cell membrane. Sometimes you'll hear this called the plasma membrane, and more on that later on in this unit. All cells have this jelly-like fluid inside called the cytoplasm. All cells also have DNA. Even though a prokaryotic cell doesn't have a true nucleus, it does still have within it DNA because it needs to have hereditary information to pass down to its offspring. And finally, all cells are going to have an organelle known as the ribosome. Now, ribosomes are not membrane-bound, so we're not violating that rule that prokaryotes don't have membrane-bound organelles. And we will talk a little bit more about the structure of the ribosome later in this presentation. So you have a diagram on your Power Notes sheet. Let's take a look at that and make sure that you have this filled out. So hopefully you were able to figure out from the image on the screen here that this larger cell is a eukaryotic cell and the smaller cell is the prokaryotic cell. And then there are certain things that they all have in common. These 3 blanks here represent things that a prokaryote and a eukaryote have in common. First of all, the squiggle here, it's inside of a structure in the eukaryotic cell, but it's just floating in the cytoplasm of the prokaryotic cell. That would be the DNA. We have this second line is pointing to that fluid interior. That would be our cytoplasm. This third line is pointing to the outer coating, which is going to be our cell membrane. Now, these 2 lines at the top are unique to eukaryotes. So what are they going to be? Well, clearly we have a nucleus here that's containing the DNA. And then these other ovals just represent membrane-bound organelles. So we can see that on this slide. If we go back to the slide, You can see all of these membrane-bound organelles. We can see a nucleus. We have cytoplasm in both cells. We have this membrane around both cells. And so that's how it relates to the diagram that we just looked at. Now you may be wondering where these complex cells came from, especially if we just said in the cell theory, all cells come from existing cells. Well, when we're talking about the origin of new life or new types of cells, this is really an area that scientists are studying but don't have any definitive answers. One of the best theories that we have to describe the origin of eukaryotes is called the endosymbiotic theory. And in this theory, we have an ancient cell, an ancestral prokaryote, has DNA inside of it, and then if the plasma membrane starts to fold in and form a protective layer around that DNA, that would be a very early form of a nucleus. And then we're talking about millions of years passing here. But over time, if this cell were to engulf an aerobic bacterium, just meaning a bacterium that's capable of cellular respiration and can make lots of ATP, it could also engulf a cyanobacterium, which is prokaryotic algae, so it's photosynthetic. If both of those events were to happen and the larger cell didn't digest the endosymbiont, the little thing that came in, then it's actually beneficial for both. These aerobic bacteria can then have a protected place to live, and they're making ATP that the large cell can use. Same for these primitive chloroplasts over here. The cyanobacteria would become chloroplasts later on. If they're photosynthetic, they're producing sugar, which the big cell can use, and they also have a nice protected place to live. So in science, every time we have a theory, we know that there has to be evidence to support that theory. This is not some wild guess. So some of the evidence that we have for the endosymbiotic theory, you have three things listed here. That's pretty compelling evidence. And it mostly has to do with the mitochondria and chloroplast. Those are the most studied organelles. First of all, both of them have double membranes. That makes sense if we think about if you're engulfing a cell, that original cell would have had its own membrane, plus surrounding it with the membrane of the larger cell would result in a double membrane structure, and that's exactly what we see in both mitochondria and chloroplasts. Mitochondria and chloroplasts also have their own DNA and ribosomes inside of the organelle. This has nothing to do with the nucleus of the cell. This mitochondrion and these chloroplasts actually have enough DNA to function completely without any instruction from the cell's nucleus. And finally, mitochondria and chloroplasts are able to reproduce on their own. It looks very much like how bacteria reproduce. So these three pieces of evidence together make a pretty good case for the endosymbiotic theory to have been a very probable explanation for how we got eukaryotic cells. So now let's talk about some organelles in more detail. This is section 2 of chapter 3 that we're about to begin here. And before we talk about the organelles and start to fill in that chart, let's define what an organelle actually is. It's a structure within a cell that's specialized for a particular function. And we do typically have these organelles working with each other. We will talk about how something is made in one place and then it's sent to another organelle for another function. So these are highly specialized structures that are doing things that a cell needs in order to survive. We're going to start with the cytoskeleton. Now, the order of your chart on your notes organizer is the exact same order of this presentation. So if you can't tell from the picture in your chart what organelle it's talking about, it is exactly the same order that I will be discussing them. So cytoskeleton is that very first organelle on your chart. You can see that the cytoskeleton, it is pretty much what it sounds like. Cyto, C-Y-T-O, is a prefix that means cell. And then we have the word skeleton. So what does a skeleton do in our bodies? It gives us strength and support. It maintains our shape. It allows us to stand up straight. That's pretty much what the cytoskeleton does inside of a cell. It can also help position and transport organelles. Organelles don't just sit there within a cell, they do move around, and they can use the cytoskeleton kind of like train tracks to be transported from one side of the cell to the other. The cytoskeleton plays a role in cell division. That's one of the topics we will learn about a little bit later in the course. And it can help the overall cell move as well, not just the organelles inside it, but the entire cell can move. Our next organelle is the nucleus. The nucleus is going to store genetic information. And when I say genetic information, I'm specifically talking about DNA. Now, inside of the nucleus, there is a region called the nucleolus, and you can see that sometimes it looks like a really dark dot inside of a nucleus when you look under the microscope. All that is the region where ribosome production is happening. The ribosomes that are found outside the nucleus have to be made somewhere, and they're made in the nucleolus. And then those ribosomes, when they leave the nucleus, they're going to leave through what we call nuclear pores. So there are openings in that membrane that's surrounding the DNA because we do need to send ribosomes and RNA out of the nucleus in order for them to accomplish their function. Next up, we have the endoplasmic reticulum, which is often abbreviated ER. This is really a transport organelle. It's a system of tubes and canals throughout the cell and really allows proteins and anything else the cell makes to be able to move from one place in the cell to another. There are two types of endoplasmic reticulum. We have the rough ER, and it's called rough because it is covered with ribosomes. All of these dots that you see here that are on the ER itself, that makes it the rough ER, and it looks really bumpy under the microscope, so that's where its name comes from. This is the site of protein synthesis because ribosomes are making proteins, as we'll discuss in a minute. The smooth ER does not have ribosomes. It's not pictured here, but it looks very similar, just without the bumps on it. And the smooth ER has the job of synthesizing lipids. So when membranes need to be made in the cell and they need phospholipids, the smooth ER is making them. Next up, we have ribosomes. So we just talked about that on the rough ER. Ribosomes are actually cellular structures that build the protein. So without a ribosome, a cell cannot make protein at all. And remember, proteins are made out of amino acids. So the job of a ribosome is just to link one amino acid to the next, to the next, to the next. These are very, very tiny structures. They don't have a membrane around them, and they are found in all cell types, even bacteria. And this is a neat picture in the background. All of these dots are the ribosomes, and this squiggly stuff coming out of the dots, that's an actual protein being made. And so that kind of gives you an idea of how small a ribosome is. Now we have the Golgi apparatus. The Golgi apparatus is going to be a stack of flattened membranous sacs. What do I mean by a membranous sac? Well, essentially it's like a bubble of membrane with an interior part where something can be stored. And so in the Golgi apparatus, its job isn't so much to store things, but a protein that's been made by the rough ER is going to leave the rough ER and go to the Golgi apparatus and pass through this stack of membranes. And what happens when it's inside these different membranes is that chemical modifications take place to get that protein ready to leave the cell. And eventually the Golgi apparatus will package that finished protein and send it to the cell membrane so that it can leave. Now, what does the Golgi apparatus send proteins out in? It uses vesicles. Vesicles, here we have another membranous sac. A lot of stuff inside of the cell is just a membrane with space inside or just a place for things to be stored or collected. And so vesicles are going to be used to transport things. If proteins go from the ER to the Golgi, they move in a vesicle. If they leave the Golgi and go to the cell membrane, they're moving inside of a vesicle. So they have to be packaged up inside this membrane to be transported throughout the cell. Next up, we have the mitochondria. We've already learned about the mitochondria because we've learned about cellular respiration. The mitochondria is the organelle where cellular respiration is happening. That means it is used to produce ATP for the cell. Now, one thing that you need to keep in mind is that this is found in both plant and animal cells. It's not in prokaryotes because remember they don't have membrane-bound organelles, but both plants and animals use cellular respiration and they need mitochondria in order to do that. Next we have a vacuole. This is another membranous sac, but instead of being used for transporting, it's used for storage. They tend to stay in place and they're just storing something to be used later. This is very distinctive in a plant cell. In this picture here, this is actually a picture of a plant cell, and this large open area on the inside of the cell, that is all a vacuole that is filled with water. And that happens a lot in plant cells because it is in the center of the cell. You'll see it called a central vacuole in a plant. Other cells do have vacuoles. If it is a small single-celled organism that is eating food, it will have small food vacuoles that it can store those food particles in until it digests it. But in terms of a defining characteristic, a really large central vacuole is a characteristic of a plant. Next, we have a lysosome, another membranous sac, but this time it specifically contains digestive enzymes. Those enzymes can break things down, whether it's old cell parts, maybe there's a damaged organelle that the cell needs to break down and recycle. This can also be used by unicellular organisms if they are digesting their food because it contains digestive enzymes. So anytime something needs to be broken down in a cell, a lysosome can fuse with that thing and release its digestive enzymes and break that thing down. Next, we have centrioles. Centrioles are going to be unique to animal cells, and they have a very important role in cell division. We have a cell division unit a little bit later on in the course, and we'll talk about spindle fibers separating chromosomes. So when we get to that unit, remember that it's the centrioles that are producing those spindle fibers. This is not something that's found in plant cells. They can undergo cell division, but they don't have this highly organized structure that's known as the centriole. And now we have a cell wall. Cell wall is going to be a rigid outer layer. It's outside of the cell membrane. There is a membrane and a cell wall here in these cells, and it's used mostly for structure and support. This is something that we do not find in animal cells. Our cells don't have cell walls, but a lot of other cell types do, like plants, bacteria, and fungi all have cell walls to help support those cells. And now we have a chloroplast. Chloroplast you have also already learned as the site of photosynthesis. This is going to capture light energy. It's going to convert it into stored chemical energy of sugar. Now this is going to only be found in things that are photosynthetic, like plants or algae. This is not an organelle that you find in animal cells. So that was our tour of organelles, and what I'm going to leave you with here today are 2 section quizzes so that you can test yourself, see what you have learned from this presentation. Section 3.1 was all of the stuff about the cell theory and the development of that and the development of our knowledge of cells, prokaryotes versus eukaryotes. And then 3.2 is all about those cell organelles that we just filled in that chart for. So go ahead and try to answer these questions. Write down your answers on your notes organizer so that we can discuss them in class. Hopefully you found this presentation to be helpful, and if you have any questions, please make sure you ask me so that I can get them answered.